All the progeny, F1e first filial generation, had the appearance of one parent only. For example, in a cross between peas having yellow seeds and peas having green seeds, all the progeny had yellow seeds. The trait that appears in the F progeny is called dominant, whereas the trait that does not appear in F is called recessive. The meaning of these results became clear when Mendel set up genetic crosses between the F1 offspring. These crosses gave the important result that the recessive trait reappeared in approximately 25% of the F2 progeny, whereas the dominant trait appeared in 75% of these offspring. For each of the 7 traits he followed, the ratio in FZ of dominant to recessive traits was always approximately 3:1. When these experiments were carried to a third F3 progeny generation, all the F2Ps with recessive traits bred true, produced progeny with the recessive traits. Those with dominant traits fell into 2 groups. 1/3 bred true, produced only progeny with the dominant trait. The remaining 2/3 again produced mixed progeny in a 3:1 ratio of dominant to recessive. Mendel correctly interpreted his results as follows, figure 1:1. The various traits are controlled by pairs of factors, which we now call genes. One factor derived from the male parent, the other from the female. For example, pure breeding strains of round peas contain 2 versions or alleles of the roundness gene, RR, whereas pure breeding wrinkled strains have 2 copies of the wrinkledness RR allele. The round strain gametes each have 1 gene for roundness, R. The wrinkled strain gametes each have 1 gene for wrinkledness, R. In a cross between RR and RR, fertilization produces an F1 plant with both alleles RR. The seeds look round because R is dominant over R. We refer to the appearance or physical structure of an individual as its phenotype, and to its genetic composition as its genotype. Individuals with identical phenotypes may possess different genotypes. Thus, to determine the genotype of an organism, it is frequently necessary to perform genetic crosses for several generations. The term homozygous refers to a gene pair in which both the maternal and paternal genes are identical, for example RR or RR. In contrast, those gene pairs in which paternal and maternal genes are different, for example RR, are called heterozygous. One or several letters or symbols may be used to represent a particular gene. The dominant allele of the gene may be indicated by a capital letter, R, by a superscript plus R, or by a plus standing alone. In our discussions here, we use the first convention in which the dominant allele is represented by a capital letter and the recessive allele by a lowercase letter. It is important to notice that a given gamete contains only one of the 2 copies, one allele, of the genes present in the organism it comes from, for example either R or R but never both, and that the 2 types of gametes are produced in equal numbers. Thus there is a 50-50 chance that a given gamete from an FIP will contain a particular gene, R or R. This choice is purely random. We do not expect to find exact 3:1 ratios when we examine a limited number of FZ progeny. The ratio will sometimes be slightly higher and other times slightly lower, but as we look at increasingly larger samples, we expect that the ratio of peas with the dominant trait to peas with the recessive trait will approximate the 3:1 ratio more and more closely. The reappearance of the recessive characteristic in the FZ generation indicates that recessive alleles are neither modified nor lost in the FR generation, but that the dominant and recessive genes are independently transmitted and so can segregate independently during the formation of sex cells. This principle of independent segregation is frequently referred to as Mendel's first law.